Hi, my name is Quinn Powers, and this is my research project, Gastrolith Identification and Replication by Means of Artificial Digestion. The goal of this project is to bring some clarity to the issue of gastrolith identification, specifically to better distinguish gastroliths from non-biological exoliths. Gastroliths were identified as polished exotic cobbles, but there is more than one way to polish a cobble, so these polished exotic cobbles are now called exoliths, and gastroliths are one kind of exolith. But now we have the problem of identifying a gastrolith opposed to other kinds of exolith. So, I attempted to replicate multiple exolith formation environments, and then analyze the rock's surface features to see if I could find any noticeable differences. I had 5 testing groups. The first group was my best attempt to simulate the conditions inside a sauropod's stomach. I used hydrochloric acid and the digestive enzyme pepsin to recreate the chemical conditions, and a rock tumbler to recreate the mechanical. I chose pine needles as food for 2 reasons. First, the modern bird capercaillie, or the wood grouse, has a primarily coniferous diet and has more polished gastroliths than other modern birds, so perhaps the needles have some polishing factor. Second, the needles would likely make an appropriate modern analog for the diet of a Jurassic sauropod, as we have found Jurassic conifers in the fossil record. To control whether or not the needles have a polishing factor, my second group is the same stomach conditions, but with equisetum reeds as food. These reeds are also found in Jurassic Age rock, so they make a plausible control food. For my third group, to see if the food has any effect, I have a stomach with no food. For my fourth group, I just have rocks and water, so I have a fluvial analog to compare to my stomach analogs. And finally, I have my untumbled control rocks, which were left alone as they were. After the rocks were done tumbling, I imaged each group using a digital camera, a microscope, and a scanning electron microscope, or SEM. I then used the SurfCharJ plugin for the software ImageJ to quantitatively analyze the SEM images. It measured the arithmetical mean deviation, or RA values, meaning the difference between the peaks and the valleys of the image's topography. In this table, you can see each group's contents, duration of tumbling, and the mean roughness value of the group. Unfortunately, my duration values are uneven, as you can see. This is due to technical issues I had with the tumblers that unfortunately became an important factor. The lowest RA values belong to the groups with the longer tumbling duration. I didn't mean for duration to become a variable in this, but clearly it is. Another source of variation comes from the rocks themselves. As you can hopefully see from these example SEM images of a quartzite, the relatively larger bumps and divots were mostly unaffected by the tumbling. The microtopography of the rocks was smoothed out and the surface became pitted, but those are small-scale differences compared to what was largely unchanged. As you can see in this box plot, there are significant differences between samples in the same group and even between pictures in the same sample due to these features. Even accounting for outlier images that capture the edge of a sample, significantly increasing the RA value, there's a lot of variation here. This issue might have sorted itself out better with a much larger sample size, but I have to work with what I was able to get in the time that I had to get it. So, while insightful, the RA values have their problems. And where does that get us? Well, ultimately the study didn't find any conclusive diagnostic features. None of the tumbling groups create any significant difference in surface features from each other, and the RA values have their problems. If anything, they suggest that tumbling duration might be the most influential variable. So we still have that identification problem. But this means there's plenty of room for future research to be done. There's a lot of variables to be tested still, including different foods, acidity, duration, lithology. There's a lot of ways to build off of this research. I would recommend a study with a larger time span than I had, both to increase the tumbling duration and to increase the analysis time to allow for a larger sample size than I had. And if possible, I would recommend not having your equipment break on you while running the experiment because, as we found out, it can impact your results. And finally, I'd like to thank these wonderful people here for their help throughout this process. And now, are there any questions?